Right. So first off, I would like to thank the organizers for inviting me. It's an honor and privilege to be here. As a matter of fact, I was so excited, I ended up preparing a 40-minute talk instead of a 20-minute talk. But please don't worry, I've cut down my slides. So I come from a computational background, bioinformatics, data science, and David asked me to talk about new technologies for exposure monitoring. So the way I've structured my talk is, first I will talk about some of the bioinformatics and data science techniques that we've developed for mining high-resolution metabolomics data, and also for integrating the metabolomics data with other omics data, as well as exposure data. And then the last half, I'll give you an overview of some of the case studies. So these are some of the recently published studies. 2 out of the 3 are in collaboration with the Department of Defense using samples from the DoD Serum Repository. So the first one will focus on benzopyrene exposure. The second one is trichloroethylene exposure. And then the third one, which was literally accepted 3 weeks ago, is focusing on integrating the ICD-9 coding data with biomolecular data and exposure data. So first off, what is high-resolution metabolomics? So high-resolution metabolomics uses liquid or gas chromatography in tandem with high-resolution mass spectrometry. So the high-resolution aspect here is the capability to tell 2 narrow peaks apart. And the advantage of doing this is we are able to capture a broad spectrum of low and high abundance endogenous and exogenous metabolites. So this becomes very relevant in the exposome research, as we are able to pick up a lot of dietary as well as environmental chemicals in our analyses. So metabolomics, in the big scheme of things, it's the closest link to the phenotype and the clinical outcomes. And it provides a functional readout of the cellular activity and physiological status and everything that happens upstream. So the way we are using the HRM platform at Emory is, so we are trying to link the environmental exposures and try to understand the metabolic response to these environmental exposures, of what pathways, of what specific metabolites are perturbed in response to these environmental exposures. And then we are trying to link this information with the health outcomes. So this is an overview of the general workflow. The first point, what's the minimum sample requirement? So for biofluids, plasma, serum, urine, we typically ask for at least 50 microliters. For tissues, we ask for at least 20 milligrams. And then once we get the samples, we have a sample preparation procedure, standard operating procedures for sample preparation and extraction. We have 5 different high-resolution mass spectrometers in the lab. We process the samples, get the raw data, and one key or one important aspect of our platform is that we analyze every sample in triplicates. This is absolutely critical for low abundance metabolites, because if you're measuring a sample only once, it's very hard to tell whether it's a noise, background signal, or it's a true signal. But by running every single sample 3 times, if we see a reproducible signal for a low abundance metabolite 3 times, then we have high confidence that this is a reliable measure for that feature. And then we have procedures for quality evaluation of the data, for batch effect. And then once we get the processed data, then we move on to the biomarker discovery and network analysis to identify which metabolic markers are associated with the disease outcomes or the exposures. So we use a variety of univariate and multivariate methods to do this. Once we've identified a subset of metabolic markers of interest, then we move on to the pathway analysis step, where we try to identify which metabolic pathways are significantly enriched in response to these environmental exposures. And we conclude by going back to the instrument level and we have to do follow-up experiments to validate the identity of these metabolites. So we have an in-house library of more than 1,000 endogenous metabolites as well as environmental chemicals that we use for identity confirmation, and we do some other experimental evaluation as well. So that's the high-resolution metabolomics aspect. But now, over the past few years, systems medicine, precision medicine, these are the hot new topics. So we are generating data from all these different platforms, and we also have the environmental exposure data, which is impacting all these different molecular layers. And then we also have the clinical data in the biorepositories or electronic medical records. So how do we combine all this data and try to make some meaningful sense of what's the multi-scale network level organization between the clinical outcomes, the biomolecules, and the environmental data. So to do that, there are a lot of different methods that are available that allow us to do this type of data aggregation. I classify them under three main categories. The first one is pathway or knowledge-based aggregation, where we utilize the information from knowledge bases like KEGG, HMDB, and utilize information about known pathways and try to integrate the gene transcriptomics data, metabolomics data, proteomics data using at the pathway level. The second one is literature-derived associations. So here we use text mining methods and mine the scientific literature. We can mine the abstracts or the whole documents and try to identify which genes, proteins, metabolites, and environmental chemicals are co-mentioned, whether at a single sentence level, at a paragraph level, or at the whole document level. And then we can sort of establish or infer some co-occurrence or associations between these different entities. And then the final category, which has been my focus, is the data-driven integration of these different data sets. So here the idea is that we use multivariate statistical methods and try to identify co-expression patterns across these different data sets using these computational methods. So the one that we published last year, XMWAS, it's publicly available as an R package. Also, there's a web URL where you can go, and there's a interface that anybody can use. You do not have to be a bioinformatics expert to do this type of analysis. So that was one of the main motivations. 90% of the users of XMWAS, they do not have bioinformatics training. So it was designed for clinicians and other biomedical researchers to facilitate them to be able to do this type of multi-omics analysis. So the way it works is that as a user, you upload different data sets. So for instance, in this case, we have transcriptomics data set, metabolomics data set, and proteomics data set. These data sets were collected on the same set of samples. So if we have 100 samples, for the same 100 samples, we performed transcriptomics analysis, metabolomics analysis, proteomics analysis. And if we had environmental exposure data, then we can also upload that. And the way software works is that it uses partial least square regression, which is a multivariate statistical method, to try to identify which genes and metabolites and proteins are correlated in a significant manner. And once it's done that, then it generates networks of those associated genes, metabolites, and proteins. And then it clusters the network and identifies subnetworks or communities of tightly connected nodes. So I'll demonstrate in the last case study how we've used this tool for identifying communities of ICD-9 codes, metabolites, and environmental exposures, and microRNA cytokine data. So moving on to the case studies. So the first one, this was a pilot study. It was published in 2016 in JOEM. This is a collaboration between Emory University, University of Rochester, Dr. Utell was also involved, and Department of Defense. So this was a pilot study where we were given samples for about 30 subjects. So this was more of a feasibility study. The idea was that before analyzing large number of samples, can we test the methodology or the analytical capability on small number of samples to see what's the likelihood of finding or seeing anything meaningful. Dr. Tim Mallon, who's the PI from the Department of Defense, they sent us 30 unidentified human samples. So they did not provide us any demographic information or any sort of information. They just sent us those 30 samples. We analyzed the samples, which were part of the Department of Defense serum repository. So these samples, they were originally collected for mandatory HIV testing in armed forces personnel, and they were stored at minus 30 degrees Celsius. So I actually went to this repository, and the only thing I remember is that it's really cold down there. Once we got the samples, we analyzed them on our metabolomics platform, and the goal of this specific study was to try to identify which metabolites and which metabolic pathways are perturbed due to benzopyrene exposure. So, which is a carcinogenic PAH arising from the combustion of organic material, found in burn pits. And then these benzopyrene was measured using a GCMS platform. So we performed a metabolite association study with benzopyrene as our main outcome and tried to identify metabolic pathways. So here are the results. So this heat map shows the clustering of top 100 metabolic features that were strongly associated with benzopyrene levels. So this is a two-way hierarchical clustering. So on this dimension, we have clustering of the metabolic features. So we can separate them into two major clusters. This cluster is the levels are lower in these samples, which all these samples have higher benzopyrene levels. And then these metabolites on this side, they have higher levels in subjects who have higher benzopyrene exposure levels. And then we were able to cluster these subjects into 4 different clusters based on the benzopyrene exposure level. And we categorized them into high exposure and low exposure. And then here are some examples of some of the endogenous metabolites that were different between the low exposure group and the high exposure group. So linoleate, sphingosine, and cholesterol. So they all had lower abundance levels in subjects that had higher benzopyrene exposure. And we were also able to identify a set of metabolic pathways that are perturbed in response to benzopyrene exposure. The second case study, it's occupational exposure to trichloroethylene. So this was conducted in collaboration with National Cancer Institute, Dr. Nat Rathman, the PI there, and the samples were collected in a factory in China. There were 95 controls and 80 subjects who were exposed to trichloroethylene. So in this study, they used the vapor monitoring badges for tracking exposure to trichloroethylene. And again, we performed untargeted metabolomics analysis on these samples. And we performed a metabolite association study to identify which metabolic metabolites and metabolic pathways are perturbed in response to TCE exposure. So we found a set of metabolites that were around 200 metabolic features that were either upregulated or downregulated in response to TCE exposure. And again, we were able to identify a set of metabolites related to methionine and cysteine metabolism, fatty acid metabolism, carnitine shuttle, and sphingolipid metabolism. And then the next step that we did in the study was we also had information for immune response biomarkers, as well as the TCE exposure biomarkers and renal damage biomarkers. So we did an integrated network analysis to integrate the metabolites with the immune response markers, renal damage markers, and then the TCE exposure markers. So essentially, the key aspect of this type of methodology is that we are able to take this multi-dimensional data coming from different platforms and put it together and identify the associations between these different layers of data. So the final case study is definitely one of the most exciting projects that I've worked on in my short career. Again, this is in collaboration with the Department of Defense, University of Rochester, and Mount Sinai. So here, we performed an integrative network analysis using the clinical data. So we had ICD-9 coding for these subjects before and after deployment. We had biomolecular data, cytokine, metabolomics, microRNA, and we also had environmental exposure data. So just to give you a brief background of this study, so the first case study was a pilot study, but then we got funded, and we were given samples for 400 military personnel from 2 time points, pre and post deployment. So again, these samples are from the Department of Defense Serum Repository. 200 of these 400 personnel, they were deployed to either Balad in Iraq or Bagram in Afghanistan. So this is our case group. And then there were 200 non-deployed service personnel who were stationed domestically. So this was our control group. And the controls were matched with the cases. So they were age, gender, and gender matched. And then, as I mentioned, the samples were collected at 2 time points, pre and post. And again, just like the previous example, these samples were originally collected for the mandatory HIV testing. So here's just a brief overview of the study. So for both groups, the samples were withdrawn pre-deployment or at the first time point. The cases, they were deployed, and post-deployment, a second set of sample was withdrawn. Those who were not deployed at the same time point as the case group, for those individuals as well, a second blood draw was performed. So in this case, the example that I'm showing, we are only focusing on the case group. Since we are interested in linking the biomolecular and exposure data with the ICD-9 outcomes, we were limited by N. So when we looked at the ICD-9 codings for these subjects, in different categories, in some categories we only had 3 subjects or 5 subjects. So what we ended up doing was we ended up focusing on this category, cardiopulmonary symptoms, which we were able to classify about 49 different ICD-9 codes under this major category. And that gave us 45 cases that developed a cardiopulmonary symptom during or after deployment. And we had 25 cases that did not develop at any point, either before or after deployment, any of these cardiopulmonary symptoms. So in this analysis, we use the 41 deployed personnel who developed cardiopulmonary symptoms and 25 who did not. So as I mentioned, we had multidimensional data available for these subjects. So we binned the data into different categories. So we had a molecular data bin, which included the metabolomics data, microRNA data, cytokine data, and the protein biomarkers. So that's matrix 1. In the second matrix, we had the environmental exposure data. So we used the first three principal components of the dioxins. We had to do that because of missing values. We had cotinine levels, and then we had measurements for benzopyrene diol epoxide. And then the third dimension was the health outcomes. So as I mentioned, we had data for 49 different ICD-9 codes for each one of these subjects. So the subjects were coded 0 or 1. So it was a binary matrix where 1 meant that the subject was given or assigned that ICD-9 code. 0 means that they were not assigned that ICD-9 code. So we did some pre-processing for this ICD-9 matrix and identified principal components of these ICD-9 codes. So the idea for doing this was because what we noticed was that when a patient is assigned a code, these symptoms are not independent of each other. They are overlapping. So for the same disorder, you can have multiple ICD-9 codes. So the idea was by doing this type of dimensionality reduction, we actually were able to, in an unsupervised fashion, cluster all the related ICD-9 codes on one dimension. So dimension 1 was primarily related to cardiovascular symptoms. So it included chest pain, palpitations, abnormal ECG, EKG, and those symptoms. Dimension 2 was primarily focused on asthma, so shortness of breath. And then dimension 3 was upper respiratory symptoms. So we took all this data and plugged it into our data integration workflow, and we got this pretty picture, which is sort of hard to interpret, but I'll walk you through it. So in this network, there are 3 types of nodes. We have the squares, which represent the biomolecules, metabolites, microRNAs, cytokines, and proteins. We have triangles that represent environmental exposures, the principal components of dioxins, BDPE, and cotinine. And then we have these circular nodes that represent the ICD-9 principal dimensions. And then these edges, they represent positive correlation or negative correlation. And the colors in this network, they represent communities or clusters of tightly connected nodes. Oh, sorry. See, I'm getting too excited again. So again, in an unsupervised fashion, we were able to identify these clusters of different heterogeneous data sets. So this is interesting, this is great, it's very impressive, but the question comes, so what? What does this tell us at the mechanism level? So what we can do is, we can take the nodes in each one of these communities, and we can run pathway enrichment analysis to identify, for instance, which metabolic pathways are associated with each one of the ICD-9 dimensions, with the environmental exposures with cotinine and the dioxins and BPDE. So this is a bubble plot. On this dimension, we have metabolic pathways that were significantly associated with either one of the ICD-9 dimensions or one or more of the environmental exposures. So we are able to see that some of the metabolic pathways, they are common between the ICD-9 dimensions as well as the environmental exposures. So for instance, alkaloid biosynthesis is associated with ICD-9 dimension 1 related to cardiovascular symptoms, as well as dioxin PC1 and PC2. So basically what we've done is, again, we've gone from this multi-dimensional data which is sitting in data files. We've performed this analysis, we've identified the associations, and we've taken those associations to a mechanistic level, at the molecular level. So we've gone from the exposures to molecular pathways. So these, I'm just only showing some of the examples. Over the past few years, we published many similar studies where we've used a combination of high-resolution metabolomics with data science capabilities to identify metabolic pathways associated with air pollution exposures. And then for the DoD study, the first supplement was published in 2016. So there are a lot more studies, very interesting studies in these supplements. And then the second supplement, it's currently in press, so it should be available sometime next month. So to conclude, the overarching goal is to link the environmental exposure data and identify metabolic response to the exposures, and then to link the metabolic response to health outcomes. And what we've done over the past 10 years is to develop analytical plus a computational framework that allows us to take the information from 50 microliters of serum samples and run through our computational workflow and integrate that data with other exposure, clinical, and multi-omics data, and begin to answer a lot of these exciting questions. So with that, I would like to acknowledge members of the Clinical Biomarkers Laboratory, Dr. Dean Jones. He's the director of the Clinical Biomarkers Laboratory that performed the high-resolution metabolomics analyses in all these studies. I'd also like to acknowledge collaborators in the DoD project, Dr. Utel, Dr. Mallon, Pamela Krall, Julie, Colin, Thomas, and other collaborators. And also the funding that we received from DoD for this work. With that, I will take any questions and comments. Thank you.
Thank you, Karan. I'm glad I didn't have to give that presentation. Well done. Questions, comments? It's, you know, there clearly are limitations to what you can do, because some of it is knowing when the exposures occurred and the timing of the blood draws. So a lot of this is the integration over time as well. The nice feature is you had the pre-exposure samples, but the drawing of the post-exposure could be quite variable.
Yeah, thanks for that. So, you know, in your first two case studies, you have a well-defined, well, in this case, the endpoint is the biomarker, right? The TCE, or the benzopyrene.
Exactly.
So you're able to use partial least squares for that.
Right.
And in our setting, we kind of like that. I mean, we have a very complex exposure, and, you know, we could potentially have biomarkers of exposure for that. But if you have a very poorly characterized exposure, is there any role for that? You know, you would then have maybe endpoint would be exposed, not exposed.
Right, right.
Which would be very, very crude in our setting, whether there would be any utility for that, you know, approach to look at different pathways involved, metabolic pathways that might be involved with that sort of scenario.
Right. So, so one of the neat things about using HRM. So, for instance, in this TCE study, the metabolomics data, it includes if you have benzopyrene, if you're exposed to benzopyrene, chances are, and then it also depends on the half-time of the chemical you're exposed to. But presuming that at the time of the collection, those biomarkers are still present, have not been excreted out, the data will capture the downstream metabolites of those environmental chemicals. So you might have a poorly uncharacterized exposure, but by doing this type of analysis, you might see signals in the metabolomics data as well that might complement the data that you have from other platforms.
I mean, in our case, we do have, I mean, there are instances where they have bloods, that are before and after, or maybe even more proximal to the exposure that we could exploit for that.
We call it suspect screening. So the idea is that, just like the case you mentioned, where you just like to take an already analyzed data set, let's just say metabolomics data set, and you want to see in that data set, were any of the samples exposed to TCE, for instance? So what we can do is, in this already collected data set, we can go in a retrospective fashion and we can do a targeted search for TCE and TCE-derived metabolites. Because we saw that in this study, there was a chlorine 3537 isotopic pattern for the TCE metabolites. So the idea is, and so you can do that in a computational way, and if you get any hits, then you can go back to the instrument or for further validation.
So, Sari, where this seemed to me an ideal technology was in the days we were doing chamber studies, where you gave someone ozone, for example, or nitrogen dioxide, whatever you were using, and then collected samples at different time points, either during exposure or post-exposure, but you really knew the timing and the concentration of the exposure. That was kind of an ideal situation if you had this technology back then. But as you say, the mix is uncertain, and the timing is uncertain. But the pathway analysis is from my perspective, quite interesting. Yeah, Cecile.
Okay, thank you. Thank you.
The Ballard-Bogler kind of choice here. Can you talk a little bit about that? Because it may be that one of the more interesting choices would be to take people who are deployed to a burn pit containing area and compare them to those, in terms of their metal, their bubble pods.
Right.
With those who were deployed to a base where there wasn't a burn.
Right. So honestly, I'm not the best person to answer the choice for Ballard versus Bagram. Dr. Till, you might have some insight on that.
The choice was that of DoD, and they provided the samples, and it was a mix. I don't know that. Initially, as the study was designed, there was kind of a separation between the two. Over time, it became, just as you said, it might have been interesting to look at these separately, and we could still do that, although the numbers, I can't remember what
I think
very weighted.
Right, they're not balanced in terms of. So I believe it's Bagram. We actually, in the second supplement, we do have a manuscript where we've separated the two out, but we are looking at the data in a very targeted fashion. So we are only looking at about 200 environmental chemicals that we validated, as opposed to doing a global untargeted analysis. Just because of the small n, we don't want to overinterpret anything. So yeah.
Yeah, you obviously there's a challenge in looking at these historical exposures, and you're left with the persistent chemicals to look at, as far as is there an impact versus something like TCE in a factory that they're using. So I guess in a similar way, if you could do a mixture of both, go back and look at burn pits in a certain way, to look at, again, who was or was not proximity to a burn pit kind of thing. And then a future effort, of course, would be to look at other endpoints of exposures to less persistent chemicals in kind of somebody that's deploying now kind of approach. I mean, I guess, do you have any thoughts on that?
No, absolutely. So honestly, so we did the first pilot study, and again, I'm coming from more a computational data science perspective, and I'm answering this. So we did the first pilot study. No one had any idea whether we'll see anything, let alone identifying biomarkers or pathways. So that was the first challenge. And then in the second one, it was a second step in the direction where Dr. Mellon and the other PIs, they designed the study in a way so that there's a maximal chance of seeing anything and being able to do statistical comparisons by doing a case group and a control group matched. But again, there are limitations in terms of the deployment. So we're still, this is still an ongoing collaboration. There's still a lot of ideas, and we all still are actively involved in this study. So I'm certain that the next study that we design will try to address these limitations.
Yeah, without going into a lot more detail, I mean, what we've learned is you need to have a much better handle on the timing of when the samples were drawn. I mean, you know, there's certainly unknowns as to how much time someone spent around the burn pit in different areas. We did some. Phil Hopke did some exposure assessment, and part of the base there was a lot of jet fuel. I mean, there are all of these kinds of different variables that began to unfold as we were doing it. If you had to do it over again, you'd do it a little differently for sure. But it is a technology that. As you refine the exposures, you really can begin to understand some of the molecular pathways in terms of, and it isn't necessarily even disease. I mean, it's just, what we were looking at were some symptom reports, generally not diseases so much. Shortness of breath, cough, were the way the symptoms were reported. But it's a next step as the exposure assessment gets better. You're certainly I think you'll be able to employ some of this technology.
Could you associate, instead of symptom scores, instead of symptom scores, could you associate markers of chronic inflammation and use those as the comparison with the exposures?
So we did that. I mean, then, you get into what are the right markers, and we looked at a variety of inflammatory cytokines. MicroRNAs turned out to be particularly interesting in terms of showing some consistent microRNAs, but as we thought this through, we were particularly interested in seeing if we could go from the exposure to linking it with the ICD-9 codes. But we've done some of the cytokines, and the question is, did we pick the right ones? Because there are lots of them, and you can run large batteries, and then you wonder if you got another 800 bloods, would you see the same biomarkers? And that's, if you ask me, the thing that I would most like to do is see if our initial findings actually could be confirmed. That's probably a key issue.
One of the charges of this committee is to look at exposure outcome relationships, and I wonder if you could just tell me I'm getting some sense of it, but tell me in a nutshell what these approaches provide above and beyond mechanistic insight. that might help us in that exposure outcome linkage?
All right. So I guess you get the information at three different levels. You are able to identify individual metabolites that are strongly either going up or going down in response to those exposure biomarkers. Going a step above, you're also able to identify the pathways. That's the mechanism part. But in the long term, especially with the third case study with the ICD-9, where we were able to identify which metabolites, microRNA, cytokines, proteins, and environmental exposures are strongly associated with onset of cardiovascular symptoms. You can design now targeted therapies. So this is getting into the precision medicine that we had a small number of samples here, so obviously we can't really claim any of the precision medicine aspect here, but what we've done with this is it's a proof of concept study that by doing this type of data integration, you're not only able to identify individual markers, you're also able to identify pathways that could be targeted for therapeutic reasons.
So Sverre, just taking it a little different in answering your question. I mean, most of these endpoints that might be, quote, disease, okay, take place over time. And so what I thought was interesting and yet to be proven is, do these signals that you're getting changes in metabolic pathways, ultimately, are they forerunners of people who have been put at risk for certain kinds of disease? And, first of all, you have to know if the disease itself actually follows these pathways. And then what is the relationship between finding these changes and disease development or symptom development over time? But to answer your question, I think in and of itself, it doesn't tell you the answer. I say in and of itself right now, it doesn't tell you the answer. It says there are pathways that have been changed, presumably as a result of exposures that took place. And
so you're arguing basically that we're searching the holy grail for the subclinical markers that are going to predict disease.
Well, I know better than to hope for the holy grail. But can I, as these metabolomics expand into other areas of disease, do we eventually find that there are these common pathways that are leading to some of, be it respiratory or other system diseases?
So a lot of the outcomes, I agree. There's some of them are sort of chronic, but some are not. I mean, and we can take the World Trade Center experience as an example, where we have some hints of some things happening fairly, not just symptoms, but illnesses that occur that are not are typically chronic diseases that are also in play here in our discussion.
Exactly. There, of course, you had that really high dose early on, or high concentration exposure, so it's different. But again, as we begin to understand how to use these metabolomics and how to interpret whether they are going to be predictive, I think it's a strategy in terms of understanding links between exposure and outcomes that has potential. Biomarkers have been around in terms of concept for a long time, and we still have trouble linking biomarkers with absolute progression.
I guess to add to that, so this can also be used for risk stratification. So you have a personnel who comes back from service you run a metabolomics analysis, you see that their metabolic profile is consistent with previous 1,000 personnel who ended up developing a cardiovascular symptom in the next 9 to 12 months. So rather than waiting till they develop those symptoms, you can act right away and put that person in the high-risk category. So risk stratification is, I guess, another application of this type of approach.
We'll have nobody participate.
All right.